Today, my partner and I would like to present our video with a title HIV Combination Antiretroviral Drug Therapy CART. However, before we proceed, what is CART? Let me introduce first what is HIV. What is HIV? HIV stands for the Human Immunodeficiency Virus. A person who is infected with HIV is said to be HIV positive. Once a person is HIV positive, that person will always be HIV positive. All viruses must intact living cells to reproduce. HIV takes over certain immune system cells that are supposed to defend the body. These cells are called CD4 cells or T cells. Here is the figure that shows the structure of HIV. The first one is HIV capsid, the HIV core that contains HIV RNA. The next one is the HIV envelope, which is the outer surface of HIV. The next one is the HIV enzymes, the proteins that carry out steps in the HIV life cycles. The next one is the HIV glycoproteins, the protein spikes embedded in the HIV envelope. And the last one is HIV RNA, HIV's genetic material. And this figure shows the entry and intracellular transport of HIV that replicate in the nucleus. Next, let's proceed to the life cycle of HIV. First, HIV attaches to a cell, then HIV will empty its contents into the cell. The HIV genetic material, RNA, is then used by the reverse transcriptase enzyme to build HIV DNA. Next, viral protein R forms membrane channel to allow the HIV DNA to pass through into the cellular nucleus. After that, the HIV DNA is inserted into the cell's chromosome by the HIV integrase enzyme. This establishes the HIV infection in the cell. When the infected cell reproduces, it activates the HIV DNA, which makes the raw material for new HIV viruses. Packets of material for a new virus come together. The immature virus pushes out of the infected cell in a process called budding. Later, the immature virus breaks free of the infected cells. Lastly, the new virus matures, raw materials are cut by the protease enzyme and assembled into a functioning virus. What is antiretroviral therapy? Antiretroviral therapy, or ART, are medications that treat HIV. The drugs do not kill or cure the virus. However, when taken in combination, they can prevent the growth of the virus. So is HIV disease, and this is known as Combination Antiretroviral Therapy, or CAT. Antiretroviral drugs are referred to as ARV. Combination Antiretroviral Therapy, CART, is also referred to as highly active antiretroviral therapy, HAART. The CART treatment involves 2 to 3 different kinds of drugs will be given to those with HIV. This is because if only one drug is given, this may provide a chance for virus particles to become resistant towards the drug. Hence, researchers decided that giving 2-3 drugs at once are sufficient to suppress the activities of virus particles and prevent them get resistant to any antiretroviral drug. This antiretroviral drug is not a cure for HIV, but is important for one from getting really sick with HIV. Besides, it can also reduce the amount of HIV to undetectable and hence reduce the transmission of HIV to someone else.
Antiretroviral drugs can be classified into 5 major categories, in which each of them inhibits a specific stage in the HIV life cycle. There are fusion inhibitors, CC chemokine receptor type 5 antagonists, reverse transcriptase inhibitors, integrase inhibitors, and protease inhibitors. So this picture shows the mechanism and site of actions of the antiretroviral drugs. The first one is the fusion inhibitors that prevent the fusion and entry of the virus into the targeted CD4 cells. Second is integrase inhibitors that block HIV integrase enzyme to prevent insertion or integrations of the viral DNA into the DNA of the host CD4 cell. So it inhibits HIV from replicating. Next, nucleoside reverse transcriptase inhibitors incorporate into the HIV DNA to stop the building process. This results in incomplete formation of new viral DNA and no virion is produced. The agents work similar to nucleosides, but they have a non-peptidic chemical structure. The fourth one, non-nucleotide reverse transcriptase inhibitors bind to viral reverse transcriptase to prevent synthesis of the viral DNA from cellular RNA. Eventually, this will stop the formations of the HIV. These agents are called as non-nucleoside inhibitors because they are not nucleoside analogues, although their mechanisms are the same as the nucleoside analogues. Lastly, protease inhibitors work at the last stage of the viral reproduction cycle by preventing assembly and release of the HIV from the infected CD4 cell. CD4 T cells is the marker to indicate the immune system level. So when the CD4 T cell count is less than 500 cells per cubic millimeter, highly active antiretroviral therapy (HAART) will be given. HAART will also start be given when the viral load is more than 100,000 copies per ml of blood. However, this HAART is given right away regardless 350 to 500 cut off of the pregnant woman, young children, and those with opportunistic infections. Jee Jap Meng et al. Claim that HIV infections is possible to be treated by using antibody-based strategies. The approaches can be thus classified as follows. First, antibodies blocking infection. Second, antibodies potentiating CD8 plus T cell responses. And lastly, antibodies directly targeting and depleting CD4 plus T cells harboring replication competent HIV-1. Okay, so the first one, antibodies blocking infections. The studies using bird neutralizing antibody (BNAbs). BNAbs target regions of HIV-1 EMV protein, but only generated in a small fraction of HIV-1 infected individuals during the natural course of infection. BNAbs are clinically proven to suppress viremia by targeting the CD4 binding sites and carbohydrate-dependent epitope in the ventriloop of the HIV-1 envelope spike. However, human studies have clearly demonstrated that BNAb monotherapy is suboptimal to achieve prolonged suppression in viremia in the absence of ART, and also associated with the rapid emergence of BNAb-resistant viruses. The second strategy is antibodies potentiating CD8 plus T cell responses. CD8 plus T cells in HIV infections are functionally exhausted and express several co-inhibitory molecules. The use of anti-PD-1 apps in HIV infection has the primary objective of preventing rapid exhaustion of HIV-specific CD8 plus T cells, but it may be induced by standard T cell activation and proliferation, subsequently leading to clonal expansion of HIV-infected CD4 plus T cells. And lastly is the antibodies directly targeting and depleting CD4 plus T cells harboring replication competent HIV-1. A study has discovered an antibody that combines three different BNAbs and known as tri-specific antibody. This antibody is able to protect monkeys from SHIV, which is a type of HIV in monkey. The tri-specific antibody binds to three different critical sites on the HIV surface. A safety trial is expected to begin in healthy people in late 2018, and a separate study in people living with HIV is still under consideration.
In conclusion, three major objectives of immunological therapy are to restore immune function, enhance the efficiency of ART in suppressing virus replication, and serve as an intervention approach that results in viral suppression in the absence of conventional ART. No effective cure currently exists, but with proper medical care, HIV can be controlled. The medicine used to treat HIV is called antiretroviral therapy or ART. If taken the right way, every day, this medicine can dramatically prolong the lives of many people infected with HIV, keep them healthy, and greatly lower their chance of infecting others. However, advanced disease of CART initiation was found to be associated with poor outcomes, indicating that earlier treatment may improve effectiveness of CART. Therefore, it is important for the patients to have a detailed history taking and physical examination at least every 3 months once they have started the ART. The assessment of CD4 must be done to observe the effectiveness of the agents. Researchers have currently come up with latest intervention of ART, which is HIV vaccines. HIV vaccines have been designed to aim to elicit so-called broadly neutralizing antibodies. However, these antibodies have rare features that limit their use as direct vaccine templates because it is unlikely that any vaccine could consistently elicit similar antibodies. For the future use, this vaccine hopefully can benefit those with HIV, and it is being targeted to enter the market in 2018. That's all from us. Thank you. Here are our references.